Okay, right. Let us rock onto Electric Avenue and then take it higher. For those of you that can't hear it first, is this coming Wednesday. That would be July 29th. And I've posted some practice problems and some practice exam questions. The format will be pretty much the same as the first.
I'm the United States Surgeon General, and my family is suffering from substance use disorder.
Okay, sorry, something happened there. Okay, right. Yeah, so the format will be pretty much the same, except you will have a couple of genetics problems that are short answer instead of multiple choice, and they'll be similar, if not identical, to what you're seeing on the practice problems that I've given you. I might try to cough up just a few more practice problems this weekend, just to give you a little more, practice. Okay, so let us go ahead and cut to the slides. So we have said all I think I've got time for about Mendelian genetics and the ways that it works. And we worked out that Mendel reasoned by counting a hell of a lot of pea plants that every organism had two copies of each gene, right? And it received one of each gene inside cells that were about to divide. In case anybody missed it, chromosomes are always present inside cells, but you can only clearly see them when cell division takes place. And I promise we will get to the deeper reason as to why that's the case. But chromosomes turned out to follow the same basic rules. Your chromosomes come in pairs. Every normal one of your cells, if it's got a nucleus and isn't something like a red blood cell, every one of your cells will have 23 pairs of chromosomes, making 46 in all. And you pass one of each pair of chromosomes to each of your kids, and you get one of each pair of chromosomes from each of your parents. So genes and chromosomes turn out to be following the same rules. Chromosomes do this shuffle thing that makes them get dealt out, so one of each pair goes to each offspring. When this was worked out, everybody had forgotten about Mendel, but when Mendel's laws were rediscovered in 1900, it was very soon realized that genes followed the same rules. And by 1902, two people had proposed independently that genes were physically carried on chromosomes. Now, genes and chromosomes can't be the same thing. Pea plants, for example, have, I believe it's 7 pairs of chromosomes, 14 in all, but they've got a heck of a lot more than 7 traits. They've got a heck of a lot more than 7 genes. So a gene is not the same thing, but Sutton and Boveri proposed that whatever genes were, they were physically carried on chromosomes. That, you know, whatever a gene was, a chromosome was the physical basis of that gene, and the chromosome might be somehow carrying many genes. That caught the attention of a professor at Columbia University, the grandson, by the way, of one of the more feared Confederates, guerrilla fighters in the Civil War, probably the greatest scientist to ever come out of the University of Kentucky, Thomas Hunt Morgan, who got interested as a professor at Columbia University in New York City. And he and his students began studying genetics using fruit flies, Drosophila melanogaster. These are the critters that will rapidly swarm in your kitchen if you make the mistake of leaving a banana out for too long. Or, you know, if a bowl of fruit on the kitchen table begins to get just a little bit overripe, these are the flies that you'll see getting drawn to that overripe fruit. So why did he study Drosophila melanogaster? Well, the reason is they're a lot easier to take care of than elephants. Mendel's laws and this chromosome stuff basically apply to all eukaryotes, but some eukaryotes are very difficult to work with. Mendel's laws apply to elephants, but elephants require huge amounts of space and food, and gestation in an elephant is 18 months. So if you crossbreed two elephants, it takes you that long to get any results. Fruit flies are a lot smaller. They're cheap to raise. All you need is discarded fruit from the corner fruit stand or whatever it is. Back in the day, they used to raise them in glass milk bottles, you know, which are mass produced and cost almost nothing. They don't take up much space. They're easy to handle. They breed very rapidly. One generation takes 21 days, I think. Or no, I think that's a lifespan. One generation might only take about 7 days from mating to new adults. So you can run, you know, 50 generations in a year. And they also have lots of traits that can be recognized easily and that follow simple Mendelian rules. There are lots of genes that affect eye color, body color, wing size and shape, things like that. And they're pretty easy to recognize. And you can also tell males and females apart pretty easily with a little practice. They also have, this turned out to be useful, only 4 pairs of chromosomes. And in certain cells, the chromosomes are really easy to study. In cells of the salivary glands, of all places, the chromosomes happen to be unusually large and thick and strongly banded. So these things that look like stripy ribbons, those are giant chromosomes from a fruit fly salivary gland. So that makes chromosomes. That makes Drosophila a great example of what we call a model organism. That's a species where there's some aspect of life that happens to be very convenient to study. Fruit flies have a lot of advantages for studying genetics. They've got those banded chromosomes that are easy to see and to handle. They don't have many chromosomes. That would turn out to be useful. And they are cheap to raise. They don't take up space. Morgan's lab at Columbia University came just to be known as the fly room because it was basically a room with shelves that were full of fruit fly jars. They're cheap, inexpensive, and you can do a lot of experiments very quickly. And fruit flies continue to be used in fundamental genetics research all over the world. There are still a lot of people that do very sophisticated work on fruit flies. Even though Sarah Palin disapproves, there's an enormous amount we can learn from fruit flies and apply to other species, including our own. Anyway, one of Morgan's students, Calvin Bridges, who, if memory serves, almost flunked out of school because he didn't go to any of his classes because he was spending all of his time staring at fruit flies. And it paid off because he discovered a mutant fruit fly. We'll look later as to what I mean by mutation. If you say mutant today, people tend to think of the X-Men. Mutations do not necessarily cause severe deformity or certainly superpowers or anything like that. A mutant is just one where you get an allele changing within one generation. We can talk later as to how that happens. Mutation is ultimately the source of new alleles. And this fly that he found happened to have white eyes instead of the normal red eyes. They were lacking the red pigments that fruit flies usually have. Okay, so Bridges saves this mutant fly and lets it breed. And if you cross a purebred red-eyed fly with a white-eyed fly, the offspring all have red eyes, so red eye color is dominant to white. By the way, when you're working with fruit flies, the alleles that are very, very common, the alleles that you would see if you went out and, collected a bunch of wild fruit flies, by putting a fly trap with some overripe banana in it or something like that. Wild fruit flies that you caught would virtually all have red eye color. So red eye color is sometimes called wild type. Wild type just means the phenotype that is by far the most common in wild populations. So wild type here just means red eye color because almost every fruit fly in the world, especially a wild fruit fly, is going to have red eyes. So red eye color is dominant to white eye color. And if you cross two hybrids from the F1 generation, we would assume that they're heterozygous, and you'd get 3 red-eyed flies for every 1 white-eyed fly. And that's exactly as Mendel's laws would predict, right? So far, this looks like just another case of, a single gene with 1 dominant and recessive allele, like Mendel's purple and white flowered peas. Except all of the white-eyed flies that you got turned out to be males. And I should mention that, I think I said this earlier, but a nice thing about fruit flies is that it is, with a little practice, it's pretty straightforward to tell the difference between males and females. Males have some extra bristles on one of their legs, making up what's called the sex comb. There's some difference in color and shape of the abdomen. So with practice, it's pretty easy to tell your fruit flies apart. And all of the white-eyed flies in this cross turned out to be male flies. Half the males were red-eyed and half were white-eyed, and none of the females were ever white-eyed. The only way you could get a white-eyed female would be to cross a white-eyed male with a hybrid female, an F1 from the previous cross, in which case a quarter of the offspring would be white-eyed females. But if you cross a white-eyed female with a red-eyed male, you only get red-eyed females and white-eyed males. Huh? It's okay if you didn't catch all of that the first time. What the heck is going on? You can clearly see that there's some kind of bias in this trait, in that it tends to be, it doesn't follow an even sex distribution. With a lot of Mendelian traits, males and females are equally likely to express a particular trait, but this one has some kind of sex bias in its expression. What is going on? Okay, this may be the missing piece that you need to do Lab 5, by the way, because we haven't quite gotten to this in lecture, but it does come up in Lab 5. And some of you may already know about this, but in case you didn't, we said that in diploid cells, chromosomes come in pairs. That's not always entirely true. In many organisms, one pair of the chromosomes varies depending on the sex of the organism that has them. In most mammals, females have 2 copies of a large chromosome that's called the X chromosome. Males, however, have 1 X chromosome and 1 different looking chromosome that's called the Y chromosome. Now, that's not universal in the animal kingdom. In birds, for example, it's females that are XY and males that are XX. In a lot of reptiles, there are no sex chromosomes, and sex is determined by the temperature at which the egg is incubated. If you raise an egg at a slightly higher temperature, it'll turn male, and at a slightly lower temperature, it'll turn out female. That's true for sea turtles and for crocodiles. Probably true for lizards as well. I'd have to look that up. I'm not 100% sure. And then a lot of marine fish are just hermaphroditic, and they'll all start as one sex and then switch to the other, or sometimes they're both at once. Yeah, life gets weird. But in mammals, the usual situation is females have a pair of a big chromosome called the X chromosome. Males have one X and one different looking chromosome called the Y chromosome. By happy coincidence, fruit flies happen to do the same thing. So there you have some chromosomes from a female human. You can see that these have been stained so that you can see the banding patterns on them. And you have 22 pairs of non-sex related chromosomes, and non-sex chromosomes are called autosomes, by the way. Everybody has those. All people have chromosome 1, chromosome 2, ... all the way to chromosome 22. And this female human has one pair of X chromosomes, and you can see they're fairly large. There they are, down there at the lower right, and they're pretty big chromosomes. And every body needs at least one X chromosome. If you don't have at least one X chromosome, you will never survive. There's the chromosomes from a male human, and 22 of the 23 pairs look the same, except you have one X chromosome down there at the lower right, and one tiny little thing called a Y chromosome down there. Without going into too much detail, you could think of the Y chromosome as being acting like a little switch. How do I want to get into this? And if so, how much? Human embryos all start out with basically the same plumbing, with external genitalia that look more or less female, although not exactly, and internal genitalia that, let's just say there's several different tubes going in different directions. If there's an X chromosome present, some of those tubes enlarge, or if there's two X chromosomes present, some of those tubes enlarge and others of them shrink, and you end up with, if everything goes as it usually does, you end up with female anatomy for the reproductive organs. If a Y chromosome is present, that's a kind of switch that causes a different set of tubes to enlarge and other tubes to retract, and some changes to happen in the external genitalia and give you your basic male anatomy. The Y chromosome you could think of as acting like a little switch that essentially says, okay, cause such and such parts to grow and such parts to retract, and this person will end up with a penis and testicles. If there is no Y chromosome, what normally happens is that the X is that you get female anatomy. The X chromosome actually does a lot more than just female anatomy. In fact, everybody needs to have at least one X chromosome. You can't live without it. But the Y chromosome you could think of as the throwing a switch that causes you typically to develop male bits. Right. I can finally talk about a female scientist at this point, just because until we're up to about the late 19th century, women have been strongly discouraged from studying science. Not that they couldn't, but they just weren't allowed to at most places in Western Civ. One of the early generations of women who were actually able to get education in science and become professors in the US was Nettie Stevens. who had been teaching at Bryn Mawr College in Pennsylvania, where Morgan had worked before he went on to Columbia University in New York City. And Stevens, unfortunately, died fairly young. She didn't get to contribute as much as one might hope, but she'd made the discovery that many insects have chromosomal sex determination, including fruit flies. Females have two X chromosomes, males have an X and a Y. That's not true for all insects. Ants, bees, and wasps, for example, do something quite different. But it happens to be true for fruit flies and for mammals, including humans. So good to know there. That's an important discovery because it tells you chromosomes have something very distinct to do with phenotype, that what chromosomes you get determines a major part of what your phenotype is. By 1911, Morgan had hypothesized that the explanation for the results he was getting crossbreeding his white-eyed fruit flies was that genes had to be physically carried on specific chromosomes, and specifically the gene for eye color was physically located on the X chromosome. That's a phenomenon called sex linkage. And in case you're bored, bear in mind that this applies to human biology as well. We've got a lot of genes that are sex-linked, and I'll show you some in a bit. We can set this up with a Punnett square. Instead of writing specific, abbreviations for alleles at the top and the left, we can write chromosomes at the top and the left. The female parent has two copies of the X chromosome, so she can pass on either one X or the other X. The male has an X and a Y, and he will pass on either his X or his Y, not normally both. Occasionally it happens, and when that does, you get offspring with extra sex chromosomes. There are humans that can be born XXX, XXY, XYY, things like that, which is interesting and has some interesting effects on phenotype, but we don't really have time to get into that. What you can do to handle this is write gene symbols as superscripts. So I've represented red eyes with a plus sign, and plus is usually the way that we abbreviate wild type. The white eye gene is carried on the X chromosome, and I've written that as X with a superscript W. And then very few genes other than that switch for sprouting male anatomy are carried on the Y chromosome. So the Y chromosome doesn't have anything to do with eye color, so we just write plain old Y. So the original white-eyed fly was a male, so his genotype must be X carrying the white-eyed allele Y, right? X with a white-eyed allele and then a Y, because he's white-eyed, but he's also male. If you mate that male with a red-eyed female, this is what you get. You get half your offspring are red-eyed females that are heterozygous for white eye color, and half your offspring are red-eyed males that are not carrying the white-eyed allele at all. Now, let's say we cross two of these together. If you cross a male from the first cross and a female from the first cross, you get this. 1/4 of your offspring get the wild type X from mom and the wild type X from dad, and they are red-eyed females. 1/4 of your offspring get the white-eyed X from mom and the wild type X from dad, and they are red-eyed females, but they're heterozygous. 1/4 of your offspring get the wild type X from mom and the Y chromosome from dad, and they are red-eyed males. And 1/4 of the offspring get the white-eyed X from mom, and the Y from dad, and they are white-eyed males. That explains why you've got that odd pattern where if you do this cross, half of your males are white-eyed, half of your males are red-eyed, and none of your females ever have white eyes. That's why you get that bias, because the allele for eye color is physically located on the X chromosome. And so males and females do not have equal probability of getting it passed on to them. Notice also that females can be homozygous or heterozygous. Males, the technical term is hemizygous, because a male only has one copy of the X chromosome. Whatever alleles he's carrying on his X chromosome, he will show in his phenotype, right? A male that has the white-eyed allele doesn't have the possibility of getting a dominant red-eyed allele to cover up that white-eyed allele, because the male can only have one X chromosome. If he's normal, you can have XXY males. They're typically sterile. That's not the way things normally go. Okay, so whatever the male inherits on his X chromosome, he's going to show in his phenotype. He can't hide a recessive with a dominant because there is no other copy of that gene. Okay, right. Well, one of the upshots of this, by the way, is that it's the male parent that determines the sex. You read sometime, I think it was Henry VIII, who wanted to get a divorce from his first wife because she only produced daughters and she didn't give him a son. Well, the fact is it wasn't her fault. It was for whatever reason, the sperm that he produced that made it to her egg cells were all carrying an X and not a Y. So it's actually the father that determines, although father can't do it consciously, but the father determines the sex of the offspring in humans and in fruit flies. This is a human example. This is a diagram that's used to diagnose red-green color blindness. People with this can see some color, but they can't distinguish red from green. If you are not red-green colorblind, you should easily be able to see there's a number 12 up there. If you are red-green colorblind, you can't see the difference between the red dots and the green dots, and this is just going to look like a ball made up of a bunch of dots. Now, red-green colorblindness is pretty common. About 7% of all men in the United States have it. Typically, if I'm teaching 3 or 4 sections of Biology 1400, I'll have at least one guy who has it. It's not uncommon at all, but it's much rarer in women. And the reason is that if you're a woman, you have 2 copies of the X chromosome, and you have to have both of your X chromosomes carrying the allele for red-green color blindness. If you're a male, you only have 1 copy of the X chromosome. So if your X just happens to be carrying the allele for color blindness, you'll be colorblind. You can't hide that recessive allele with a dominant allele because you're only going to have 1 copy of the allele. And red-green color blindness follows exactly the same rules as white eye color in fruit flies. I'm obliged to mention cats, of course, as I seem to be turning into a cat person. There's one sleeping right at my elbow as I'm talking. An example is the gene for orange color. It's dominant to the gene for no orange color. And incidentally, because of a type of epistasis, if you have a dominant gene for orange color that's present, that gene also blocks the action of the gene for black and brown color. So this orange cat right here could be carrying genes for black, medium brown, light brown, dilute, whatever. And you can't tell because the gene for orange color is blocking the expression of the genes for black and brown color. It's another case of epistasis. We don't really need to worry much about it here. But the gene for orange color happens to be sex-linked. A male cat is either going to have an X with the orange gene and a Y, which will make the cat orange, or X without the orange gene and a Y, which makes the cat not orange. It'll be black or brown or gray or whatever it might be. So a normal male cat will either be orange or it won't be orange, but female cats can be heterozygous. And in this case, for interesting reasons that we don't really need to get into, but you can think of this as a type of incomplete dominance, a heterozygous female cat will have patches of orange mixed with patches of not orange fur, like this cat here. This is called tortoiseshell. So that's a tortoiseshell cat, or a tortie, if you'd rather. And you see this cat has patches of orange and not orange fur. And it's possible because female cats can be heterozygous. Male cats can't. Male cats are either they're orange or they aren't. And incidentally, if you have a cat that's heterozygous for orange fur and also has that gene for white spotting, that's a calico. So this will have patches of orange, white, and not orange. In this case, it's black. And the upshot is that torties and calicos can only be female. Well, very, very rarely you'll get a male tortie or a male calico, because if there's an error in meiosis, a cat might inherit an extra copy of the X chromosome. And so you might have a male cat that is XXY, but that cat will be sterile. And that will be very rare. That's something on the order of 1 in 10,000, 1 in 20,000 cats that happens. So leaving that aside, the great majority, almost all tortie and calico cats are females. Now you know why. Okay, one more fun example before we take a break. There are lots of other sex-linked traits in humans, and one of the best known is factor IX hemophilia. There's several different types of hemophilia. One of the most common types is called factor IX, because you're missing something in your blood that's called factor IX, that enables your blood to clot. And if you have this, you can lead a more or less normal life, but you have to be extremely careful because if your blood does not clot or clots very slowly, you can bleed to death from an injury that would be minor to almost anybody else. You know, even a small cut, you can lose huge amounts of blood because you can't stop the bleeding on your own. And factor IX hemophilia happens to be caused by a recessive allele that is sex-linked. It follows exactly the same pattern as orange coat color and red-green color blindness and white eyes and fruit flies. And this actually runs in the royal families of Europe. There's Queen Victoria of England. She was not hemophiliac, but she was heterozygous for it. One of her X chromosomes was carrying the hemophilia allele. Her other X chromosome had the normal allele, and hemophilia is recessive, so she was not hemophiliac herself. So she married Prince Albert. It always makes me think of the old joke where you call up the tobacco shop and say, "Excuse me, do you keep Prince Albert in the can?" "Why, yes, we do." "Well, you better let him out." Prince Albert was a brand of pipe tobacco. Anyway, hey, the funny jokes cost extra. So anyway, Queen Victoria had lots of kids with Albert, and they ended up marrying into all of the major royal families of Europe. Queen Victoria did not pass on her hemophilia X chromosome to her oldest son, Edward VII of England, and he's the ancestor of the present British royal family. So the British royals don't have any history of hemophilia. Let me see. Oh, her daughter Alice did inherit her hemophiliac X chromosome. Fortunately, Prince Albert had an X chromosome that carried the normal allele, so Alice was a heterozygote. She was a carrier, but she didn't have it herself. But one of her sons was a hemophiliac, Frederick William, and then her daughter Alexandra married Tsar Nikolai II, the last emperor of Russia. And they had 4 daughters, and we don't know if they were carriers or not. And then their son, the Tsarevich, the crown prince Alexei, was in fact a hemophiliac. And he got that trait from his great-grandmother, Queen Victoria, and just by luck, she passed on her hemophiliac allele to her daughter Alice, who passed it to her daughter Alexandra, who happened to pass it on to him. Just one of those things. It also showed up in her daughter Beatrice, who married into the royal family of Spain and passed the hemophiliac allele on to her daughter Victoria Eugenie, who married King Alfonso of Spain and had 2 sons who died of hemophilia. Well, they didn't die of hemophilia, they died of racing fast cars, but that's not really a good hobby to take up if you are a hemophiliac. Kind of a bad move there. So, yeah, there, Queen Victoria's granddaughter was a carrier of hemophilia. 2 of her brothers were hemophiliacs, and 2 of her sons were hemophiliacs. So her son, Prince Gonzalo, died from bleeding after a car crash. They are the uncles of the recently abdicated King of Spain, Juan Carlos I. He abdicated, about 10 years ago, I think. He is not a hemophiliac, but his uncles were, because they were all descended from Queen Victoria. And then Queen Victoria's granddaughter, Alexandra, is sitting just to the right of Tsar Nikolai II, the Emperor of Russia. She married him, and they had 4 daughters, and I can never remember all of them, but the youngest daughter was Anastasia. There was an animated film about her that you might have seen at some point. There was Anastasia and Olga and Tatiana and the one I can't remember. And we'll never know if they were carrying the hemophilia allele or not, because when the communists took power, the entire royal family got shot. So they were shot and their bodies had acid dumped on them and were buried in a pit. So I don't think we know whether they were carrying hemophilia or not. But we do know that the crown prince, Alexei, who was in line to be the next emperor if it hadn't been for the glorious October Revolution of 1917 that kicked out the royal family and installed the communist government. If it hadn't been for that, Alexei would have been the next emperor, and he was a hemophiliac. And just by dumb luck, he had inherited Queen Victoria's X chromosome that was carrying hemophilia, that was carrying the recessive hemophilia allele. Wasn't anything anybody did. It wasn't anything anyone planned for. It was just one of those things. And yeah, unfortunately, he was executed with the rest of his family in 1920. Incidentally, Alexandra, as a mother, of course, she really wanted to treat him and, give him some, ease and comfort. And she sought out the services of a spiritual healer whose name was Rasputin. And if you're of a certain age, you will now start singing "Ra Ra Rasputin, lover of the Russian queen." Rasputin probably did not actually have a love affair with Alexandra, but he was suspected of it. But he became very close with the royal family because they believed he had healing powers that would ease Alexei's hemophilia. So there, now you know. Anyway, Morgan and Calvin Bridges and another guy, Alfred Sturtevant, went on to demonstrate that genes are also carried on autosomes. Remember, that's any chromosome that isn't a sex chromosome. And I'm not going to have time to go into the nuts and bolts of how this was done. But Sturtevant was able to locate where genes are on chromosomes, and he was able to do it with a nifty trick called linkage mapping. Remember that during meiosis 1, right? Meiosis is how the fruit flies would make gametes, right? How they make eggs or sperm. And in meiosis 1, chromosomes in the same pair actually join up together and they do what's called crossing over, right? They'll actually break and swap pieces. This is also called recombining, by the way, or recombination. But this happens during meiosis 1. And Sturtevant was able to use a little nifty trick to work out how frequently recombination happens between genes on a chromosome. And the more frequent recombination happens, the farther apart the genes are from each other. I'm not going to say more than that, just that he was able to use this property of chromosomes, this crossing over in meiosis 1, in a clever way to localize where genes physically were on chromosomes. And he was able to construct what we call linkage maps. That is part of a linkage map of the X chromosome of a fruit fly. You can see on the right, all of these are phenotype traits. So you can see yellow body, scute bristles. I think that's bristles that are flattened. White eyes, facet eyes, echinus eyes, whatever that is. Ruby eyes, cross veinless wings, all of those. And those are the traits on the right. And then on the left, those numbers represent the location of each gene along the chromosome, measured in units called centimorgans. So there, that's what Sturtevant and others were able to do. With some very careful crossing experiments, they were able to not only conclude that genes were physically on chromosomes, but genes have a particular location along the length of a chromosome. And you can even work out what that location is. You can work out what order the genes come in on a chromosome. So that immediately raises the question of what are chromosomes made of that enables them to do that? How are chromosomes able to carry genes on them? And so I'm going to pause the presentation and stop the recording, and we'll pick this up after break. Go ahead and take, let's call it 15 minutes.